Today, we will be discussing on transformation, transduction and transfection. So, recombination is a process in which a new recombinant chromosome, one with a genotype which is different from either parent, is formed by combining its genetic material from two organisms. So recombination, it results in a new arrangement of genes or the parts of genes and is normally accompanied by a phenotypic change. Microorganisms can carry out different types of recombination. So there are very effective three modes of gene transfer that is seen in bacteria, which includes the transformation, transduction and transfection. So these methods of gene transfer, it leads to the development of molecular cloning. So the basic principle of molecular cloning is the transfer of the desired gene from the donor to a selected recipient for various applications in the field of medicine, research, gene therapy with an ultimate aim of beneficial to the mankind. So first we'll see the transformation. So transformation is the uptake of naked DNA molecule or a fragment by a cell from the medium and is incorporated into the recipient chromosome in a heritable form. So the process of transformation, it was first discovered by Fred Griffith in the year 1928. At the time of the lysis of the bacterial cell, it leads to the release of large amount of DNA into the surrounding environment. And these DNA fragments, it may contain several genes. And if these bacterial DNA fragments come in contact with a competent cell, that is, the one that is able to take up the DNA and be transformed, it can bound to the cell and the DNA can be taken inside. The frequency of transformation of a competent cell is approximately 10 to the power minus 3 when an excess of the DNA is used. Here, competency is a complex phenomenon and is dependent on the several conditions of the bacterial growth. Here, taking an example of Streptococcus pneumonia, they become competent during the exponential growth phase. That is, when the bacteria is actively dividing or when the population reaches about 10 to the power 7 to 10 to the power 8 cells per ml. When the population becomes competent, the bacteria, it secretes a protein which is known as the competence factor, and these functions to stimulate the production of 8 to 10 new proteins which is required for transformation. Artificial transformation can also be carried out in the laboratory by treatment of the cells with the chemical which is known as calcium chloride. And this renders the membrane more permeable to DNA. Relatively high concentration of DNA, that is higher than would normally present in nature, are used to increase the transformation frequency. And it is easier to transform bacteria with plasmid DNA since plasmids are not easily degraded as linear fragments and can replicate within the host cell. Now we'll come to transduction. So transduction is the process by which the DNA is transferred from one bacterium to the another by a virus. So transduction, it does not require physical contact between the cell donating the DNA and the cell receiving the DNA. Transduction was first described by Joshua Lederberg and Norton Ginder in the year 1952. So there are two types of transduction. The first one is the generalized transduction and the second one is the specialized transduction. So first we'll come to generalized transduction. This generalized transduction, it occurs during the lytic cycle of the virulent or the temperate phages. During assembly, when the viral chromosomes are packed into the capsid, random fragments of the partially degraded bacterial chromosomes also may be packed by mistake. As the capacity of the capsid is limited in quantity of the DNA to be packed, some viral DNA is left behind. Hence, the quantity of the bacterial DNA that is carried depends on the size of the capsid. And the resulting virus particle, it infects another bacterial cell but does not initiate lysis. Therefore, this phage is known as generalized transducing particle. Generalized transduction can be either complete or abortive. In complete transduction, the transduced DNA fragment, it gets integrated into the recipient bacterial chromosome, forming a recombinant chromosome. And this DNA fragment, it gets replicated along with the bacterial chromosome replication and it passed on to the daughter cells. While in the case of the abortive transduction, the transduced DNA fragments, it may not integrate it into the bacterial chromosome and it remains in the cytoplasm as the free particles and they cannot undergo replication. So now we'll see specialized transduction. Specialized transduction is also known as the restricted transduction. In this type of transduction, the transducing particle, it carries only a specific portion of the bacterial genome. Specialized transduction, it occurs when there is an error in the lysogenic cycle of the phage. So when a prophage is introduced to leave the host chromosome, excision sometimes may be carried out improperly and hence the resulting phage genome contains a portion of the bacterial chromosome which is about 5 to 10% of the bacterial DNA which is located just next to the integration site. Here, a transducing particle, it injects the bacterial genes into another bacterium. So one of the best studied example of specialized transduction is the lambda phage. So now we'll come to transfection. So transfection is the process of introducing the foreign molecule and the genetic material into the eukaryotic cell. So it is one of the method of gene transfer where the genetic material is introduced into the animal cell with an aim to study various functions of protein and the gene. The term transfection is commonly used for non-viral methods of gene delivery in eukaryotic cells. So transfection is commonly used in biological laboratories for studying modulation of gene expression, gene function, gene regulation, protein expression, biochemical mapping, mutational analysis, and protein production. So this mode of gene transfer, it involves the creation of pores on the cell membrane, enabling the cell to receive the foreign genetic material. And the significance of creating pores and introducing the DNA into the host mammalian cell contribute to different methods in transfection. Chemical mediated transfection involves the use of either calcium phosphate or cationic polymers or liposomes. Electroporation, sonoporation, optical transfection, hydrodynamic delivery are some of the non-chemical based gene transfer. Particle-based transfection uses the gene gun technique where a nanoparticle is used to transfer the DNA to the host cell. There are two main types of transfection. The first one is transient transfection and the second one is the stable transfection. Transient transfection is the transfection in which the foreign DNA is eliminated by the cell prior to or during mitosis. Thus, the foreign DNA does not carry over to the new cell and they are unaffected. Whereas, in the case of stable transfection, it is the form of transfection in which the foreign DNA, it becomes a part of the cell's original DNA by either adding onto it or replacing the old DNA to create new DNA strand. So transfection method, it can be divided into three groups. The first one is methods that rely on carrier molecules. The second one is methods that deliver nucleic acid directly to the cytoplasm. And finally, the methods that employ viral vectors. Transfection using calcium phosphate was one of the earliest methods that is developed to introduce foreign molecule to a cultured mammalian cell. In this method, calcium chloride containing Herpes buffered saline solution is used to deliver plasmid DNA. Then development of lipid and polymer based carrier molecules having the capacity to bind this nucleic acid, it led to the adoption of these compounds in transfection. Liposomes that are formed from these compounds are able to fuse with the cellular membranes by which they can deliver this bound DNA or the RNA to the new cell. The transfection reaction is commonly performed under aqueous condition. So some of the problems that are associated with the traditional transfection method like calcium phosphate co-precipitation, DEAE, Drexten, polybrin and electroporation, including low efficiency of DNA delivery, poor reproducibility, cell toxicity and inconvenience. So the cationic lipid reagent mediated transfection, it yields a high and previously unattainable transfection efficiencies in a wide variety of eukaryotic cells. So with cationic lipid reagents, the DNA solution is not deliberately encapsulated within the liposome. Rather, the negatively charged DNA, it binds spontaneously to the positive charged liposomes and forms a DNA cationic lipid reagent complex. So transfection of adherent cells using various agents such as lipofectamine, it requires optimization of the amount of plasmid or the reagent and cell number and specific for cell type. So these molecules, they can work in a number of different ways to carry DNA into the target cell and insert it into their genome. The typical process involves mixing the desired DNA with the reagent, adding it to the cell in the culture, incubating them, and then identifying them which contains the new DNA. Then the use of these viral DNA vectors as well as the mechanical methods like microinjection, where the desired DNA is directly injected to reduce the risk of degrading the material. So gene guns are another popular tool for DNA transfection. So that was about the transformation, transduction and transfection. And I'll conclude today's discussion by adding that bacterial recombination is a one-way process in which the fragment of DNA is transferred from the donor to a recipient and is integrated. So there are very effective three modes of gene transfer seen in bacteria, which includes the transformation, transduction and transfection. So this transduction is the transfer of bacterial genes by the viruses, and transformation is the uptake of naked DNA molecule by a competent cell and is incorporated into the genome, while transfection is the process of introducing this foreign molecule and the genetic material into the eukaryotic cell. Finally, the transfer of desired gene from donor to a selected recipient has a wide application in the field of medicine, research, gene therapy, which aim to benefit the human mankind.